Okay, so let's start from the beginning. All right, so this is muscle, first lecture for test 3. And so if this is under your microscope, say, what tissue is this? Be specific. You would go skeletal muscle. Notice that it's striated. It has these little stripes, right? There's a dark stripe, a light stripe, a dark stripe, a light stripe, all the way down. Really, really distinct looking. Has a nucleus there. I think you can see one there. Maybe on the other side there's one there. So it's multinucleated. So this is a skeletal muscle fiber. Fiber is the same thing as cell when you're talking skeletal muscle. Fiber equals cell, right? So I could go true or false. Skeletal muscle is multinucleated. True. Skeletal muscle is involuntary. That would be false. It's voluntary for the most part. True or false? It's striated. You go true. All right. On cardiac muscle, just know the basics. It's singly nucleated, according to your book. It is striated and it's involuntary. Don't forget you got intercalated disc in there too. Smooth muscle, involuntary, single nucleus, singly nucleated, no striations, hence the name smooth. So probably not going to be showing you smooth muscle or cardiac muscle on this test because we'll get to those on another test. Okay. Let's concentrate on skeletal muscle. It's called that because mainly it moves the skeleton by pulling on tendons that are connected to the bones. There's exceptions to that, like around the upper part of your esophagus is skeletal muscle that helps, bring the food down. But for the most part, yeah, skeletal muscle is hooked to the bone, hence the name skeletal muscle. Functions: locomotion, maintain posture, support soft tissues, regulate orifices like around your Oris, right? You can purse your lips, right? Maintain body temperature. You get cold, you can shiver, and that'll keep your core temperature up for a while until you run out of ATP, of course. All right, here's just some terminology. Sarcomere is the plasma membrane of muscle cells, right? So it's just another name, right? Anytime you see sarco, think muscle. Sarcoplasm, same thing as the cytoplasm of the muscle fibers. Remember, fiber cell, same thing. So that's basically the space inside. Skeletal muscle fibers have multiple nuclei. That's because there was a bunch of young ones called myoblasts, and they fused together to make one long fiber. And for some reason, they leave their nuclei behind. Why? I don't know. That's just the way it is. So anytime you see sarco or myo, as a prefix, think muscle. All right, so artists love this because they can draw it almost exactly like it is. So here's a drawing. Here's a real microscope slide. All right, so you can see multinucleated, striated. Beautiful. Now then, let's zoom in a little bit, and let's see what's inside of here. So we're going to zoom in even more. Okay, here. And so you see these little cylindrical areas inside of here that are packed full of, guess what? Contractile proteins. Myosin, they drew in blue. Actin, they drew in red. Actin and myosin, I'm sure you've heard of that somewhere down the road. But this little bundle right here is called a fibril. F-I-B-R-I-L. Now, yeah, technically it's a myofibril, and the fiber is a myofiber, but I'm kind of lazy. I just go fibril. If you do that on the test, you're going to get credit. And then the little actin and myosin, those are filaments. Yeah, technically they're myofilaments, but if you just say filament, that's cool. I'm good with that. All right. Do we have a lot of mitochondria? Yes. Mitochondria is plural. Mitochondrion is singular. Then what's this blue stuff that's wrapped around your fibrils? That's sarcoplasmic reticulum. You go, that kind of sounds familiar. Is it anything like endoplasmic reticulum? Yeah, same thing. It's just that in muscle, the endoplasmic reticulum, which is called the sarcoplasmic reticulum, has a specialized function in addition to any other functions, and that is to store and release an ion. What is that ion? Calcium, and we'll explain why later. All right, look at these tubules that are going in a transverse direction across here. So those are called T tubules. T standing for transverse. You don't even have to spell that out. You don't have to say transverse tubule. If you say T tubule, I'm good with that. Okay. So we'll talk about the function of this in a little bit. So there's my sarcolemma, by the way. Yeah. Okay. So lots of mitochondria. There's my fibril with a bunch of filaments inside. There's my sarcoplasmic reticulum. There's my T tubules. Okay, everybody's cool? All right, so actin. So myosin is considered the thick filament. So when the light's shining up through your microscope slide, it's going to block out more light. So that's going to make it appear dark. So those dark stripes, remember your striations, that's mainly myosin because it's blocking out the light. Actin, more light goes through and around it, and that's considered the thin filament. It's actually a little more on the actin, but traditionally they say actin is the thin filament, but really it's some other stuff on there, and we'll talk about that in just a minute. And all of it together is called the thin filament. Okay, so sarcoplasmic reticulum should be an equal sign. Equal. Sarcoplasmic reticulum equals the endoplasmic reticulum in muscle fibers. It releases calcium for muscle contraction. Terminal cisternae. Cisternae is plural. Cisterna would be singular. That's the widened ends of the sarcoplasmic reticulum. You can kind of see them here a little bit. See the way the artist tried to draw this where it looks bulged out? It's kind of like bulged out right there and right there. Those are terminal cisternae. Okay. Transverse tubules we talked about. And the triad would be 2 terminal cisternae plus the T tubule. So in other words, if I made a bracket like I do here on your test, and I go, what are these 3 things together called? All you got to do is say triad. You know, I love it when they name stuff like that. You just go triad, you get the point. Okay? Now, let's look at it a little closer up. So here's a fibril. Don't forget we got more than one fibril inside of there. Right? And you can kind of see the actin and the myosin, but it's kind of covered up by this sarcoplasmic reticulum. And there's a nice looking T tubule. So you're just seeing it a little bit blown up here. And there's your sarcolemma. Now, zoom in even closer and you look on the myosin and you go, whoa, it's almost like little golf club heads on here. And then look, it looks like it's kind of connecting to the actin. Not always, but sometimes it does, right? And so something's going on here. And so hence, they kind of figured out the way this stuff works, and it's the sliding filament theory of muscle contraction. Oh, here's a T tubule. You can see it's an actual tubule. So the T tubule carries a signal. So basically, we'll talk about that in a little bit. So say if your axon is hooked to your sarcolemma through, a junction like a synapse, and then basically the signal's coming down your axon from your neuron, and it's not showing it here, but it's going to send a signal down this membrane and then a signal down this T tubule, and then that signal is going to cause this sarcoplasmic reticulum to release its calcium. We'll get to that. Don't stress on it right now. All right. But here's what I was talking about. Whoop. So there's the axon. Okay. There comes a signal. This is a motor neuron. Probably, let's just say it's in your spinal cord, right? And it's sending a signal out here to a muscle fiber. And here's my little synapses. Remember synapses from 101 or 130 or whatever it was you took? And from that little synapse, you're going to spill some neurotransmitters into that gap. Synapse is like the little gap. Okay. They call this the motor end plate on the other side of this gap. And in this situation, and so basically you're going to have neurotransmitter come out here, and the neurotransmitter is going to do its thing, and we'll talk about that in just a minute. But it's going to hook in, it's going to basically bind to receptors. Okay, and then those receptors are going to be, could be like little channels, and they're going to open, and then something's going to happen. So we'll talk about that in a minute. All right, so first of all, let's see, how is a fiber directed to contract? So here's, remember I said, oh, let's just act like that motor neuron's in the spinal cord. Well, here they do use more than one. And there's an axon coming down. See, they colored him in red. And then it's showing the fibers that it's innervating or controlling, drawn in red. Okay, so that's the ones that are going to respond if a signal comes down that axon. Then they did the same thing with the blue, and it's controlling the blue ones. And they did the same thing with the purple. They're just showing you the different fibers that these control. Okay, and so all the muscle fibers controlled by a single motor neuron, in the skeletal muscle example, is called a motor unit. And that gets really important in physiology too, folks. Okay? So motor units can be small or they can be large. So if this is controlling like 2 cells, 2 fibers, or 4 fibers, or 6 fibers, or whatever, that's pretty much small motor unit. If it's controlling 1,000, if one axon is controlling 1,000, and that's possible, fibers, then that's a large motor unit. Okay, so the sarcomere, functional unit of skeletal muscle. So if I just ask that question, what's the functional unit of skeletal muscle? Boom, sarcomere. I'll show you what a sarcomere is in just a second. Sarcomeres are connected in series to make myofibrils. You can have a lot of sarcomeres inside of a myofibril. It says here possibly up to 10,000. Each sarcomere contracts, shortening the length of the entire myofibril. Kind of like the, I guess these are supposed to be like sarcomeres. Okay. All right. So here's a fibril, and we're looking at it. We go, oh, okay. So this line that looks like a Z in this actin right here is called a Z line. This line is a Z line. So from Z line to Z line, that's a sarcomere. Technically, it's a Z disc because you see this thing's like a cylinder, but you can't see that when you're looking at it under a microscope. It just looks like a line, so they call it a Z line. Okay. Now, all this myosin right here, that they call the A band. See the A band? Where the actin, that's mostly actin, there's some other stuff too, right? The light stripe right here, they call the I band. Okay, so in other words, this dark one right here would be an A band. That would be an I band. That would be an A band. That would be an I band. That would be an A band. Okay, so that's what you're seeing. Okay, so remember sarcomere, Z line to Z line, but you got way more. See, these things just keep on going all the way down the length of that fiber, right? So it could be lots and lots of sarcomeres inside your fibrils. Okay, this line in the middle, guess what? They call it the M line. Okay, so we got I band and we have A band, and then we have I band and A band. The way I remember it, A has the letter, dark has the letter A in it, and light has the letter I in it, right? That just helps you remember which one's what. So don't forget, light is shining up through here. When you have it, say you had a fiber on your microscope slide, light's underneath it, and it's shining up through here to be picked up by your eye and the oculars. And so the myosin's going to block out more light, so it's going to look dark. Well, here's the kicker. This filament, this fibril, and that fibril, and that fibril, and that fibril, they're all lined up perfectly. So the Z lines are all lined up with each other. The M lines are all lined up with each other. The Z lines are all lined up. So they're all perfectly aligned, each of them. So that means when the light shines through, it gives you that perfect pattern. Dark, light, dark, light, dark, light. Now, had they not been lined up like that, they never would have figured out this sliding filament of muscle contraction for years and years and years, right? This gave them a clue, and I'll show you how that works in just a minute, right? So, remember we got almost like little golf club heads on our myosins? Well, hold on, let me shut that off. All right, I'm back. So, they almost look like little golf clubs. See the way they have a shaft and a little head? Actually, these are two heads, but you can't really see it the way they drew it, right? And so the little heads of the myosin can hook to the actin, okay? So the actin, they drew like these little ping pong balls. Those are the little globular actin molecules, right? And they're, they make a strand like this. Now, this myosin really wants to grab one of those actins if it can. But look, something's in the way. What's this rope-like thing that's in the way of my binding sites? They did the binding sites in yellow. That's troponin, tropomyosin. You just got to remember that. So troponin is this part, and the tropomyosin is the rope-like part. Okay? So that's in the way. So this little head cannot cross-bridge and bind to the actin as long as that's in the way. Well, this little head can do a couple of motion, right? It can cock and bind, and then it's got this hinge region, and then it can do a power stroke. So it can pull on that actin. But don't forget, you got bunches of them that can do this. So see, these guys are all bound, right? And they can pull, and they can pull this actin towards the middle, right? And so the Z line is going to go that way. And this Z line is going to go that way, right? So in other words, your sarcomere is going to get shorter. But did my actin and my myosin and my actin actually change lengths? No, they're just going to slide past each other. Hence the sliding filament theory of muscle contraction. Okay, so don't forget this stuff's all connected. Okay, and so if all these little myosins pull on these actins, it's going to make this sarcomere shorter. And then this sarcomere, same thing, all the way down, right? So that whole muscle fiber is going to contract, okay? Well, guess what's going to happen? You see the way it's kind of clear inside the middle of your A band here? That's because there's no actin overlapping. Well, right here it's not, but once you pull them in, it does. So that clearing in the middle is going to get dark, okay? And then this, where your eye bands are, these are going to get skinnier because it's pulled in, right? So this was their clue that something was sliding, is because when they looked under the microscope, fiber in a contracted state versus a non-contracted state, they saw some differences, okay? So it should be on this slide here. Here's a contracted, this is the same sarcomere. It's right, but these guys pulled in, right? So they can pull them in, and then they can let go, they can grab another spot. So that's called cycling. So it does this a lot of times, it's kind of like a little ratchet, and then it pulls these guys in. And as it does, there goes your actin, and it fills in this clearing in the middle. And so this is called the H zone. They now call it the H band, I still like calling it the H zone, but that H zone goes away because look what's happening, or at least gets skinnier, because the actin is sliding in. So see the way these guys pulled them in, right? They're taking turns. There's thousands and thousands of these things, and they're all cycling at different times, right? Some are still attached so that it doesn't slide back out, while some of them pull, and then they all take turns, basically, okay? So when that clearing right there goes away, then your H zone gets tiny, or it goes away completely, and your sarcomere gets shorter. True or false? Filaments, actin and myosin, get shorter during a muscle contraction. False. They just slide past each other. The sarcomere itself gets shorter. So let's take a look on that very first slide to see if we can see an H zone. Probably should be able to. Oh yeah, this is about the best light microscope you can get. See how it's kind of clear right in the middle? That's an H zone. Your Z line, which I can't really see it here, but they would be here. Be a Z line there, and a Z line there, and a Z line there, right? So that was their clue. Whenever they looked at it in a contracted state versus a non-contracted state, they had differences. You know, your H zones went away, your I bands got tinier, but your A bands stayed the same, ironically, right? Because the myosin's still the myosin. It's just sitting there, right? Doing its thing. You know, the little heads are bringing the actin in, but the actual links of the myosin, you know, they stay the same. The links of the actin stay the same too. It's just that they get pulled in, right? So hopefully that makes sense to you. Let's look at this a little closer. There's a couple things we didn't talk about here. Well, one thing anyway. See this green stuff? This is called titin, T-I-T-I-N. I think it's one of the largest molecular weight proteins in the body. So it's kind of like a slinky, they think. Remember the little toys you played with when you were a kid? Like a little curlicue thing? So they think it just holds the ends of the myosin so they don't flop around, right? So they do bunch up, right? Where actin and myosin don't bunch up, they slide past each other. But this will bunch up like a big spring or a slinky, right? So that's why they got it drawn like this. Okay, there's your M line, by the way. Okay, just don't miss the point and name stuff, right? Sarcomere versus A band versus H zone versus I band versus your proteins, which would be myosin. It's your thick filament. Actin, which is basically your thin filament. Your titin, right? And your M line. So don't forget the point and name stuff. Okay. Now, technically, all of this is your thin filament. That's what I was telling you about. It's a little more stuff than just actin. But traditionally, when somebody says, what's a thin filament? Somebody writes actin, you know, they get credit. But it's a little more stuff on there, okay? Did that. This is getting a little too deep. This is getting into physiology territory, but I'll just give you a basic thing here. But here goes our neurotransmitter, right? So in this case, it's acetylcholine. That's what that stands for. That was the first neurotransmitter discovered, by the way. Otto Lowy, 1923, I think it was. Could have been 22. I can't remember. Look it up. So say the acetylcholine goes across, it can bind to like a receptor that happens to be like a channel, and it's gonna, this channel, say it's a sodium channel, that's gonna open and let sodium flip across this membrane, because you got a lot of sodiums out here extracellular, right? And so you got a concentration gradient, right? You don't have as many sodiums on the inside. So there's a polarity, or there's a difference, right? And so if you open a sodium channel, that sodium's going to flow inside, okay? down its concentration gradient. Well, what's going to happen, you're going to get positives on the inside, and you're going to change this electrical polarity of this membrane. Don't stress on this stuff too much. Just try to get the basics, right? Well, that's a signal, right? So that changed this membrane, potential, they call it. And there's little gated channels all the way down, and it's kind of like that. When this flips, and these positives go over here, it's almost like a field of magnetism, or electric field almost. And that's going to cause other channels to open, and that's going to cause more sodium to go across. And then you're going to get this sodium going down here, over here, over here, and that's basically your signal. So it's kind of the same way that an axon works. So you'll learn about an axon on next test. And so basically you're going to send that signal down here. Don't worry about the DHP receptor, none of that. You'll learn that in physiology. But the end result is, once this signal goes down the T-tubule, see T-tubule, it's going to open this little gate right here, and the calcium is going to flood out. Okay, so that calcium is going to flood out. Okay, so don't forget the big picture here. Your signal came down from your neuron, down this axon. Neurotransmitters were released that cause these little sodium channels to open, and that sent your signal, as more and more of them open, down your T-tubules. Now, in the skeletal muscle, it's important to note this acetylcholine is not leaving this gap. It's not flowing down the T-tubule. I can go, true or false? Acetylcholine flows down the T-tubule. False, right? That acetylcholine is in this gap doing its thing with these receptors. And once it does it, boom, it gets broken down really fast. There's an enzyme in here that breaks it down. And I think they have it written here. I thought they did. Acetylcholine esterase. And that will break down that acetylcholine really fast. Okay? So basically, then you'll quit sending a signal if that breaks it down enough and quickly enough, and all of it. Okay? Now, what's the goal? We're trying to get the calcium out here. Why are we trying to get the calcium out around the filaments out here? Because the calcium, remember the troponin, tropomyosin? They're calling this the troponins. They drew it a different color. The calcium, they think, binds to the troponins, which causes the troponin, tropomyosin to shift out of the way. When it gets out of the way, guess what? Hey, my myosins can hook on and do their power strokes. If ATP is present. You need ATP to do that, right? Because that's an energy using thing, reaction. And so that's why you got so many mitochondria scattered around. Okay, so look what happened. Calcium flooded out, it bound here, and the troponin, tropomyosin moved out of the way. Now my myosin is like, hey, my binding sites are open. Boop, 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 and they can do their power strokes. And then what's that going to do? A bunch of these guys are doing it at the same time. It's going to pull the Z line this away, and that's going to pull the actin this away. And then this would be your H zone right here, so it's pulling it in. There's your M line, by the way. So hopefully that makes sense. And this is just kind of, and we'll shut this down after this. We'll break this into 2 30-minute lectures. But here it is, and just kind of briefly done for you, because I could go describe the sliding filament theory in your own terms, in your own words, from contraction to relaxation. And this is kind of broken down into 10 steps. So, first of all, your action potential came down and boom, acetylcholine's released, right? Then what happens? The signal goes down the T-tubule. You don't even have to say all that stuff about sodium channels and all that. Just go, signal goes down the T-tubules, right? That's number 2. And they call that signal the action potential. When a signal's coming down an axon, they call that an action potential. Okay, awesome. Now, it's going to reach the sarcoplasmic reticulum. What's going to happen? Boop, calcium's going to flood out. When the calcium floods out, you might want to give a little more info here, but it's basically active site exposure because the troponin, tropomyosin gets out of the way. And then my myosins can bind. What's that binding? It's cross-bridging over here, so they call it cross-bridging. It's crossing over that gap and bridging to the actin. So that's called cross-bridging. Now that's not the pull. It's not pulled yet. That's just the cock. Remember I said it can cock, and then it can do the power stroke. So this the myosin, the little head right there, will flip up, cock right there, and cross-bridge. Okay. Now it's going to pull, and that's called a power stroke. And then that's the contraction is beginning. So don't forget, a bunch of these things are doing this at the same time. And it'll pull, and then it'll let go. And then it'll grab in a new place and pull again, let go, grab in a new place, pull again. But you got thousands of these things doing this at alternate times. And so that's called cycling, when it's cycling like that. Okay. Now, don't forget to say contraction happens using ATP. Don't forget to say it uses ATP. How do you stop it? So that's the contraction. Whoop. How do you stop it? First, you got to quit sending the signal. So your brain says, okay, I don't want to contract that muscle anymore, right? If it's a voluntary thing going on. And then guess what? You quit sending acetylcholine across. So does the acetylcholine esterase eat that acetylcholine? Does it break it down really fast? Yes, it does, right? By the way, the choline gets recycled, I think, and they use it again after that gets, another acetyl put on it in here. So now signal's no longer going. We're no longer releasing calcium. Guess what? We're taking calcium up. How in the heck are we taking it up? Well, there's little pumps that run all the time, and those little pumps are pumping that calcium back up. Well, guess what? That calcium is going against its concentration gradient. You don't have that much out here. You're putting it back up in the sarcoplasmic reticulum where you have a lot, and then that's that uses ATP because you're going against your concentration gradient. Now, all your calcium went away, so what happened? Whoop, you can't see it, they didn't draw it here, but your troponin and tropomyosin went back in place, and now you've blocked all your binding sites. So no more cross-bridging, right? No more cross-bridging, the contraction ends, and the Z lines get further apart. So that's it in a nutshell, from contraction to relaxation. So just go over that until you kind of got it figured out in your brain. But this, and we'll stop right here, but that's just showing you the myosin binding to the little binding sites and pulling. And it's showing you, I know it's hard to see, but you see the little yellow ball that's going down there and hooking onto the purple? That's your calcium binding onto the troponin, and the troponin tropomyosin is getting out of the way. A couple of things on here that we didn't really talk about. Well, we did a little bit on one of them. That's ATP, the green that's in the head of the, I don't have any way of stopping that, but it goes in, there's a pocket in the little head of the myosin, and the ATP fits in there. Well, there's enzymatic activity in that pocket so that it can break it down into ADP, an inorganic phosphate. So that's what you're seeing happen. You see it breaking into two pieces. And don't worry about the little cone of gray. That's magnesium. We didn't talk about that, but you need magnesium to do a lot of different enzymatic or a lot of different actions. So I think it's used in something like 400 plus different reactions. So it's an important mineral. Okay. All right, so let's shut her down there, and then we'll come back. And so I'll go ahead and put this up on YouTube for you, and then we'll do the part 2. Okay.